This is our first lecture on cells, structure, and function. Here are your learning objectives for this lecture. Primarily, we're going to be going over the different organelles inside the composite cell, describing their major functions. So, why do we study a composite cell? What is a composite cell? It is a hypothetical cell structure that includes many of the common cell structures and organelles. And the reason we do this is because the cells of the human body, the 260 different varieties, are very different, and their form is related to their function. So if you look at this first one here, this is a neuron or a nerve cell, and it is designed specially to transmit signals, okay? So it has a unique structure for its particular function versus an epithelial cell, which is sheet-like and forms layers of tissues, versus a muscle cell, which is full of these lines of contractile proteins so that the muscle can lengthen and shorten. So all of these cells have distinct features and distinct functions, but they share a lot of common attributes, and that's the composite cell that we'll look at. I also want to point out the vastly different sizes of cells. So this is a human ovum or egg cell, which is really quite large, and a smooth muscle cell, also large but long, versus really itty-bitty, teeny tiny blood cells. Okay, in the composite cell, which is the general cell that isn't a real cell but contains many of the major features, you have a nucleus, cytoplasm, and the cell membrane. Those are the three major parts of the composite cell, but there are a whole bunch of organelles that you need to know a lot about too. The nucleus is enclosed by a thin membrane called a nuclear envelope and contains the cell's DNA. The cytoplasm houses all the organelles of the cell, suspended in cytosol, which is the fluid that fills the cell up. The cell membrane is also called the plasma membrane or plasmalemma, and that contains the cytoplasm. So it's the outer layer that is around the cell and holds everything together. Cells with a true nucleus are called eukaryotes. So humans, animals, anything else that has a nucleus in a cell that contains the genetic material is a eukaryote. In contrast, prokaryotes are bacterial cells. They don't have nuclei and they don't have membrane-bound organelles. And then you also have heard, of course, of viruses. Viruses are actually not alive. They're even simpler than cells. All they are is genetic information in a protein coat, and therefore they need a cell to reproduce. They can't self-reproduce, so not actually alive. So here is the composite cell. Again, it's not a real cell. It's a cell, an example cell that contains many of the features of other cells in your body. So you can see all of the major parts. Most importantly, the cell membrane, which encloses the cell, is made of this phospholipid bilayer. Okay, there's the square they've blown up. The nucleus of the cell contained in the nuclear envelope, okay, and the chromatin in there, that is the DNA, and the nucleolus is another part of the nucleus. And then everything else that's kind of filled in here, you see lots of organelles, and they're suspended in the cytoplasm or cytosol. That's where all the organelles are just kind of hanging out. So let's talk a little bit about the cell membrane, although we do this in much more detail in another lecture. It's a phospholipid bilayer made of lipids, okay, and it has heads in one side and tails on the inside, two layers oriented like that. The heads are hydrophilic, they like water, and so they orient to the outside, whereas the tails are hydrophobic, they don't like water, and they orient to the inside. And these two layers surround the cell in its entirety. Now, the most important thing about the cell membrane is this idea of it being selectively permeable, or selective permeability. Permeable means that some substances can pass through. Selectively permeable means that only some things will pass through. The cell membrane is really good at letting some things in and keeping other things out. And in that way, it controls something called signal transduction, which is basically the functions of the cell. All the things that the cell needs to do are controlled by things coming in and out of the cell telling it how to act. The cell sends messages to other cells that way. It gets incoming messages. It knows what to do because of what the cell membrane lets in and out. All right, so the cell membrane, in addition to being lipid bilayer, is home to a wide variety of proteins. So right here, this is a transmembrane protein, meaning it goes all the way across the membrane. Then there's these that stick in the membrane, and they have different functions. And again, we're going to go over this more in another lecture. But there are receptor proteins, which receive and transmit messages, integral proteins, which form pores or channels. I talked about ion protein channels, stuff that's to help things pass through the cell. There are enzymes in the cell. There are cellular adhesion molecules, which allow the cells to stick together if they wanted to make a layer or a sheet. And there are these cell surface proteins that establish self. And that's really important so that the immune system doesn't attack the cell. It recognizes that it as one of your own cells and doesn't attack it. Now, inside that cell membrane is the cytoplasm, which is networks of membranes and organelles and abundant protein rods and tubules that form a cytoskeleton. And in the cytoplasm is where the majority of the activities in the cell occur. So I'm going to go through now the organelles pretty much one by one. The first one to talk about is a ribosome. So ribosomes are produced by the nucleolus, which is inside the nucleus, and they're found on the rough endoplasmic reticulum. So this is endoplasmic reticulum right here, these lines, these layers, and the little dots on top of them are the ribosomes. And the ribosomes are really important for protein production. They are actually the enzyme that allows amino acids to be linked together to form proteins. And when we do translation, you'll get to see that in action. And since we're talking about ribosomes, we have to talk about the endoplasmic reticulum. So there's the rough endoplasmic reticulum, which has the ribosomes, which I just showed you. There's also a smooth endoplasmic reticulum, which does not have ribosomes on it. So the structure of the endoplasmic reticulum is membrane-bound, flattened sacs, elongated canals, and fluid-filled vesicles. It serves as a tubular transport system, widespread throughout the cytoplasm, and can send molecules to different places in the cell through its tubes. Parts of the endoplasmic reticulum are interconnected, and they also interact with the cell membrane, the nuclear envelope, and lots of other organelles. Involved in protein and lipid synthesis. So the one that is involved in protein synthesis, of course, is the rough endoplasmic reticulum because it has those ribosomes on it. The smooth endoplasmic reticulum is more involved in lipid synthesis. It synthesizes fats. It also absorbs fats, breaks down drugs, and is especially abundant in liver cells that help break down alcohol and other substances. And here, of course, is the analogy that I showed you, Beyonce looking like the rough endoplasmic reticulum with the ribosomes attached to it. Okay. Next organelle is the vesicle. Vesicles are membranous snacks. No, it's not snacks. Membranous sacs that form when a portion of the cell membrane folds inward and pinches off. We talked about endocytosis and exocytosis. Those are vesicles. So this one is endocytosis, and it's a vesicle being formed, going inside the cell, getting pinched off, and becoming a vesicle. These vesicles show exocytosis. They're coming down to the edge of the cell membrane, docking or fusing with it, and releasing substances to the outside. So vesicles bring material from outside the cell into the cytoplasm, and they're transport organelles. They bring substances into and out of the cell and move them around the cell. The Golgi apparatus, the structure is a flattened membranous sac called cisternae. Okay, it's right here. It's usually off the end of the endoplasmic reticulums. It looks a lot like it, but it's a little smaller. It is continually receiving vesicles from the endoplasmic reticulum. So substances go into the endoplasmic reticulum, proteins are made, they move through the tubular transport system, and they go into the Golgi apparatus. The Golgi apparatus then processes and transports the proteins as they pass through the layers. So I like to think of the Golgi apparatus as a post office. It receives incoming proteins, molecules, vesicles from other organelles, in particular the endoplasmic reticulum, and then it processes them, sorts them, and then sends them back out. So you can see the vesicles over here getting pinched off the Golgi apparatus, and they actually show it going all the way to the edge of the cell and being secreted outside the cell. The mitochondria is the powerhouse of the cell, which means it is involved in releasing energy from glucose and other nutrients and making ATP. When you take in glucose and other nutrients, your body takes it, goes into the cells, and it is turned into ATP by the process of cellular respiration. So you have to have mitochondria in order to have energy for the cell. It is a crucial organelle. People that have problems with their mitochondria have exercise intolerance, and they are weak and flaccid. Another organelle is the lysosome. This is the garbage disposal of the cell. So these organelles degrade and break down carbohydrates, proteins, nucleic acid, foreign particles, anything that has kind of outlived its worth in the body or is foreign to the body. These are abundant, of course, in white blood cells, which are part of the immune system, so they attack foreign invaders and get rid of them. Peroxisomes are similar to lysosomes. They break down lipids and biochemicals. They detoxify alcohol. They also synthesize bile acids. They're abundant in liver and kidney cells. The centrosome is an organelle that is located close to the nucleus in a cell that's in interphase, and it consists of two cylindrical structures called centrioles. The centrioles, during the cell cycle, pull apart the chromosomes and distribute them to opposite sides of the cell. So they're really important in cell division. Both cilia and flagella are involved in cell movement. Cilia move back and forth in a coordinated manner to propel the cell or to propel things over the surface of the cell. One example of this is the movement of mucus over the lining of the respiratory tract. Cilia are destroyed by chemicals in cigarette smoke, which impairs the respiratory tract's ability to expel bacteria, and that is what produces the smoker's cough. The flagella here, the only flagella in the human body, are on the human sperm cell, and they are also used to propel the cell. Microfilaments and microtubules, together they create the cytoskeleton that provides strength and shape to the cell. So they're all in that cytoplasm, making the cytoskeleton, holding the organelles, giving the cell shape. Microfilaments are rods of the protein actin. They provide for cellular movement in addition to their function in the cytoskeleton, and they also form myofibrils in muscle cells, which we will look at in great detail in a couple months. Microtubules are composed of tubulin. They're rigid, they hold their shape, and also for movement, and they form cilia and flagella and centrioles. Okay, so here's a clinical condition relating to an organelle. A child is small for her age, easily fatigued, slightly developmentally delayed. Her blood work reveals that she has elevated levels of biochemicals such as pyruvate and lactic acid that indicates that she is unable to extract maximal energy from her nutrients. So which organelle is not working properly? The organelle that supplies energy for the cells is the mitochondria, so this would be a mitochondrial disorder. Okay, the last piece to look at is the cell's nucleus. So I said before that it contains the genetic material in the cell, or the DNA. It has a few components. It has a nuclear envelope, which surrounds it, and that is studded with nuclear pores. It also has a nucleolus, which creates ribosomes, and chromatin, which is the unwound DNA. I've included this slide so you can see how some of the organelles work together. to produce proteins and lipids, in fact. The nucleus of the cell houses the genetic information, which is used to create proteins. You can see this coming out of the nuclear pore here is mRNA. We'll talk about that a lot more in a couple weeks. And it goes and uses the ribosomes to create protein. The proteins travel through the endoplasmic reticulum, and they end up going into the Golgi apparatus, and then the Golgi apparatus secretes a vesicle, which allows the protein to leave the cell. At the same time, the lipids are synthesized in the smooth endoplasmic reticulum, and they are also released outside the cell with the protein that is produced. This is the chart that's in your textbook that covers all of the cell parts that I just discussed and gives you a nice summary. And these are your learning objectives, except for this last one. You do not need to explain how particular substances will cross the membrane for this lecture. You do have to do it for the cellular transport lecture, which is next.